Good morning, good evening, and good afternoon, everyone, depending on what time zone you're coming in from. It is my pleasure to welcome you to this third day of the IHCC Symposium. So I'm Professor Nikki Mulder from the University of Cape Town. I lead H3A Bionet. Next slide, please. So it's my pleasure to just give you an outline of the events for today. So we'll start with a keynote address, and I'll introduce Ambrose shortly. We're then going to do something a little bit different. And in this speed round session, we're going to be giving you an opportunity to go to 6 different speed round sessions to discover and learn about different topics. But I'll describe that in a bit more detail after the keynote. We'll then have a 30-minute break and then come back for the final session of this great event, which is a session focused on COVID-19 and how the different cohorts have been managing the COVID response. And this will end with a fantastic keynote, we hope, by Dr. Francis Collins. And then we will end with just a short summary and sum up with the next steps from Jeff Ginsberg. So now I would like to, in the next slide, please, to welcome Professor Ambrose Wonkum. Professor Ambrose Wonkum is a professor of medical genetics at the University of Cape Town. He's a director of GenMap, which is Genetic Medicine of African Populations. And he's also the immediate past Deputy Dean of Research at the Faculty of Health Sciences at UCT. So Ambrose is PI of a sickle cell data coordinating center. He's also PI on several H3 Africa genetics-related projects and ELSI projects. He's the current co-chair of the steering committee of H3 Africa, and he is current president of the African Society for Human Genetics. So it's my pleasure to hand you over to Ambrose Wonkum, who'll be talking on why we should invest in African genomic variations. Over to you, Ambrose.
Thanks, Nikki. Thank you from just in front of your office from Cape Town. And I would like to thank the organizer to invite us and to try to share some of our thoughts on why we should invest of African genomic variation. I think this is the right forum to make the right advocacy for such a program. There are 3 major reasons why we should invest in African genomic variation. The first is ancestry, the second is ecology, and the third one is equity. Is in following these 3 points that my presentation will be laid out to you today. We will start with the missing variant that we are likely to see in African genome. There are a report from 2 years ago that using an African pangenome approach, at least 10% of the content of the African genome from a small sample of 900 African was not present in the current human reference genome. The study published from less than 500 whole genome sequence of African by H3 Africa also identified at least 3 million single nucleotide variant that was not present in the current reference genome. We know that at least 10% of variation within African genome that came from a specific humanoid that have lived in Africa at least for the past 300,000 years are not well captured and probably would be important in future with relation to health and disease. In a similar way, a certain proportion of Neanderthal DNA within the present European Asian have been associated with numerous conditions, including susceptibility to COVID-19. We know that some variants in present-day African are more current and more frequent than in European and Asian and have been important for the development for medication, for example, against cholesterol. In a similar way, there are some variants that are specific to African today that are associated to type 2 diabetes. So we have more and more variants to discover if we invest in African genomic variation. For monogenic disease that are the focus of my work, we know that there is some level of allelic and locus heterogeneity. For example, for a condition called Huntington disease, if you investigate Huntington disease at the molecular level, in 99% in European, it will be due to one gene, and the very same gene will explain only 70% of Huntington disease in population of African ancestry. The other 30% will be due to another genes. For congenital hearing impairment, our work have shown that the gene that explains nearly 50% of children that are born deaf in Europe and in Asia explain 0% in most population of African ancestry. So the African ecology that span from a north-south axis have been a motor of natural selection based on variability of direct climate, environment, and food. One example is the relation with the sickle cell disease mutation that have been selected because of malaria in Africa. Another example is variation in APOL1 that have been associated with trypanosome, and we know that that variation today is responsible for kidney dysfunction in African American, in African living on the African continent. Other environmental factors like dengue fever have allowed selection of specific variation in two genes that are present day, that are still frequent in the present day African from Latin America, for example. So I hope that in these few slides, we have shown a few glimpses to convince everyone that studying African genomics variation is scientific imperative for everyone else. I would like now to see how we can use those variations to address the issue of equity. Africa, despite the 300,000 of human genome history in the DNA, may not necessarily take advantage of that because of the low investment in innovation and wealth creation in Africa. And for the past 10 years, this has been changing by massive investment in programs such as H3 Africa that have allowed recently to have a fine mapping on genetic variation within African genome. That is allowed to development of GWise array that may be more relevant for population of African ancestry. And I'll show why this is important in a few slides. And H3 Africa also allow us to build capacity for people that would be probably handling the issue of African genomic variation in future research on the African continent. This is a small group of people trained in my own research entity. I'll now use three examples to show how we can address the issue of equity. For example, African ancestry and complex threat. Only 2.5% of participants in GY study are from African ancestry. The reason, and these GY studies do not necessarily take into account the African genomic diversity, and the reason why the polygenic risk score at moment that associates some threat to genetic variation perform very poorly in population of African ancestry, as illustrated here with data from UK Biobank. We know that we are hitting all of us, we are harming all of us by not including more African, because these 2.5% of participants are responsible for 7% of all phenotype association. And by using other approaches like whole exome sequencing, a study in schizophrenia shows that the replicated variant found in numerous genes in South Africans of African ancestry, but yield much larger effect size with 5 times less sample number in a population from COSA ancestry. I will now use another example of how we can address equity, which is the example of sickle cell disease. This is the most common monogenic recessive disease of humankind. 80% of newborns are born in Africa, 300,000 per year. This condition may be used as an example to investigate both monogenic and complex trait because it's monogenic but very frequent and subjected to genetic variation for its clinical expression. For example, there is variant, at least 3 genes that condition kidney dysfunction in sickle cell disease. We know that a variant that associated with fetal hemoglobin level have been associated with long survival in sickle cell disease. And this knowledge have allowed the development of mice model that have been cured for sickle cell disease, and recently allow the first successful gene editing for curative treatment of sickle cell disease. But the reason I think we still need to invest in fetal hemoglobin genomics as one of the way to address equity is that the genes loci that we know now explain only 20% of variation in African sickle cell disease patient investigated in Africa. And I argue that the other 80% of missing heritability can be found in population African ancestry if we use the appropriate DNA array. For example, using H3 Africa design array, we reinvestigated fetal hemoglobin genomics in Africa, and we identified at least 3 novel loci that are associated with fetal hemoglobin level. And the combination of all the loci was able to explain up to 70% variation. Two of the loci replicate very well between Cameroonian and Tanzanian and African American. So if what we found is correct, that means that this real patient with sickle cell disease will have now more option for gene editing for curative treatment of sickle cell disease. Genomic understanding of other variation, including those associated with hydroxyurea, may help find new therapeutic approaches for sickle cell disease treatment. And we know that because sickle cell disease has been on the African continent for a very long time, it's possible that Tutankhamun might have sickle cell disease based on a few studies on mummy from Egypt. It is possible that some variation might have been enriched within population of African ancestry, specifically those affected with sickle cell disease, to protect lung survivors, as illustrated by a few studies that we performed recently that identify up to 12 genes in which there is recurrent mutation, some of which are associated with lung survivors, people that are more than 50 years without treatment in Africa. And one of the genes is associated with L-glutamine pathway that we know have been recently validated by the FDA for new treatment of sickle cell disease. If we extend this study to more patients and more sites, it is possible that some of these loci that have been selected may be due to the pressure of the disease itself of the genome, may lead to new way for treatment of this condition on the African continent. And moreover, if we investigate those variants that have been concurrently selected because of numerous pressure of the environment in Africa, we may find a new way to design a genetic risk model for sickle cell disease. And this paper, we illustrated that 5 genes, for example, have been co-inherited in Africa, could be inherited by sickle cell disease patients because of the superposition of the frequency of the African continent, APOL1, G6PD, alpha thalassemia, and much recently, a variant in piezo1. The second example that I will use to how we can address equity by sequencing more African is by using genetics to investigate rare disease. I will show just one specific example here. This individual with achondroplasia lived in Africa during the Egyptian time, and that was during the 4th and 5th dynasty. This individual called Snep is the testimony that rare diseases have been on the African continent for a very, very long time, and is an area where we need to invest because this area may provide new knowledge for everyone else. And the last example that we can use to address equity, I call it the case of a silent epidemic, and this is genetic of hearing impairment. As we say in the introduction, if you pick up a child with hearing impairment at birth, in 50% of cases in Europe, this is due to GGB2 genes. In Africa, in most cases, it's not due to GGB2. And we have been investigating many Africans from at least 6 different African countries for congenital hearing impairment. Over the past 2 years only, we have identified a lot of variants in novel genes of hearing impairment. We have also shown that by investigating hearing impairment in Africa, we were able to show that maybe some of the variants are differentially frequent if we compare hearing impaired individual versus control. That shows that it is possible that some of these variants might have been selected in the population for the reason that we still need to investigate. And this knowledge will not only help development of diagnostic within the African continent, but also for population in diaspora, in addition to the novel genes that will improve our global understanding of hearing impairment pathobiology. This is, for example, in this family, with this kid with Waardenburg syndrome, we discover a novel genes. I haven't indicated which one, but I will probably share that in a publication very soon. We know that by sequencing this, we will probably contribute to address the issue of exome variation diversity that we all know are important for a disease gene pairs variation analysis in the world globally. And it's our hope that by convincing a few of the editor and hopefully people in this room that we need to sequence more African, and I propose that we sequence at least 3 million African, genome of African, to capture the whole diversity. We need to build this project based on the success of other projects that are already available. And we know that this project will not only contribute to human genetic understanding of variation, but will actually contribute to some level of economy. And we know that over the past a few years, by investing $1 for human genome, the return on the economy, for example, in the US have been 5. I don't know any single bank that can offer that level of return. So investing in African genomic variation will go above genomic and health, genomics and society, but also improve some ways of health economy globally. And I would like to thank my funders that have trusted me for some of this project, and thanks all my collaborators that have contributed to generate the data that I presented to you today. I thank you very much for your kind attention.
Thank you very much, Ambrose, for a fantastic presentation and for really giving a strong case for why we need more African data. I think this has also been highlighted through many of the discussions also yesterday about the increased diversity that's needed in genomics generally and in cohorts. So thanks very much.
Hello, everyone. I'm Francis Collins, Director of the National Institutes of Health, and really pleased to be able to speak to you as one of your keynote speakers. This one mostly about COVID-19, but I'm going to say a few other things about the IHCC, which I am such a fan of. So yes, I want to express gratitude for how this global collaboration has come together for this summit. And particular thanks to Jeff Ginsburg, Terry Manolio, Peter Goodhand, and many others who have worked tirelessly to make this possible. I also have to point out, of course, that IHCC can't operate without financial support. So a big shout out to Corey Bargman and the Chan Zuckerberg Institute for having recently made an additional contribution to IHCC, joining the ongoing support from Wellcome, from all of us, and from 7 NIH institutes, all of which makes this possible. So let me also reflect a little bit on the origins of IHCC, and then I'll get to COVID-19. The initial proposal about this kind of a cohort consortium was discussed at the HEROES meeting in June 2016. I remember that very well, the HEROES being the heads of international research organizations. And following that, with a lot of time and effort and planning, the first summit occurred at Duke in March of 2018. I remember that really well too. Since then, I've seen remarkable progress in advancing global cohorts data atlases, development of cross-cohort polygenic risk scores, and 6 other scalable pilots. Much credit to all of you. And to do so, especially in the last 16 months, with the overshadowing concerns of COVID-19, has been pretty amazing. You have now assembled over 100 cohorts in 43 countries, comprising more than 50 million participants. And that's more than an order of magnitude larger than any previous single cohort. And the potential here for global population-based research focused on precision health is really remarkable. And no one would have predicted in March 2018 that IHCC would be needed to play such an important role with a global pandemic. We didn't see that coming back then. The past year reflects, in my view, scientific self-assembly at its best to take on the COVID challenge. With rapid design, development, and manufacturing of COVID vaccines, using both conventional and novel platforms, we have made major progress with vaccines, reversing the dogma of vaccine development taking many, many years. We've shown the value of public-private partnerships in the US, our effort called ACTIVE, Accelerating COVID-19 Therapeutic Intervention and Vaccines, connecting 20 pharmaceutical companies with multiple NIH institutes, CDC, FDA, and the European Medicines Agency, has accelerated therapeutic trials for more than 20 different agents, leading to such advances as the demonstration that monoclonal antibodies can provide benefit for outpatients, not so much for inpatients, but for outpatients. And this has been happening in lots of other places. The UK, the National Health Services Recovery Trial, now enrolling nearly 40,000 patients, 10% of those hospitalized in the UK, with 13 treatment arms demonstrating the benefit of dexamethasone and of IL-6 receptor targeted monoclonal antibodies. but also in all of these instances showing what doesn't work. We need to know that too. Hydroxychloroquine, lopinavir, ritonavir, convalescent plasma actually turning out to be pretty disappointing despite what was initially a lot of enthusiasm. And WHO with their Solidarity trial, extending to 30 nations, testing anti-inflammatory agents and other matters. All of those examples of how the scientific community, and many of you have been involved in these, has risen to the challenge of trying to develop diagnostic therapeutics and vaccines for COVID-19. The All of Us study here in the US had its own response to COVID-19, one of which was a survey of 108,000 participants to see how they were coping with COVID-19. Lots of interesting data there that you are welcome to have a look at. And they tested over 24,000 samples for SARS-CoV-2 antibodies on multiple antibody platforms, making sure that this was really rigorously controlled, and identified positive samples as early as January 2020. In 5 states, those positive samples preceded identification of the first case, telling us that the virus was present sooner than we knew. And I want to give a big shout out to the COVID-19 Host Genetics Initiative, this remarkable collaborative effort to unravel the host genetics of both susceptibility to infection and severity of infection, sharing resources, protocols, and questionnaires, doing the analyses across studies to identify these determinants of susceptibility and severity, pooling individual level genetic clinical data, carrying this even beyond a straightforward GWAS, 160 registered study, and a preprint posted this month identifying 13 loci at high statistical confidence. Very impressive work. I have to wonder, looking at that, could this approach even be used to identify genetic factors contributing to some of the less common responses to COVID-19 or to the vaccines? Could this be used, for instance, to try to identify which children have come down with multi-system inflammatory syndrome, MIS-C? Which respondents to vaccines, the adenovirus vaccines, have developed this thrombotic thrombocytopenic syndrome, TTS? Is there some predictability there? What about those who have had breakthroughs from vaccines, who are fully immunized or should be, and yet still get reinfected? Is there, in fact, a subtle hint there of some immune deficiency that might be mapped using genetic approaches? And as I'll come to in a moment, what about long COVID, those individuals who don't seem to fully recover? What are those genetic factors that play out in that risk of a longer term outcome? So all of those things, I think, heightened by the success of this remarkable collaboration, could potentially be followed up on in terms of the Host Genetics Initiative. Well, COVID-19 has also reinforced several best practices, the high value of RCTs, the importance of generating diverse health data, spanning genomics, social determinants, environmental exposures, electronic health records, the holistic view of health, which these sets of cohorts are very much invested in. Now, what about long COVID? I mentioned that a minute ago. Of all the surprises that COVID-19 has brought us, this ranks very high, that somewhere between 10 and maybe as many as 30% of individuals who are infected with COVID-19, weeks later are still not returned to normal. They have fatigue, they have shortness of breath, they have brain fog, these cognitive problems, this clouding that is very frustrating to those who are so affected. And we don't understand the basis of that. We don't really understand exactly who is most susceptible. It's clear that people who are hospitalized have a higher risk of these consequences, but there are people who had mild disease treated outside the hospital who also suffer from long COVID. So what is this about, and what might we do to try to understand that? That seems like a very high priority, because this could potentially turn out to be a long tail of really serious health consequences. So how does the scientific community rise to this occasion? Well, I know all of you are thinking about that, and I think IHCC has a potentially really important role to play, given these 50 million people who are already part of these cohorts, many of whom got COVID, and some of whom probably now are affected with long COVID. In the US, we have been given by the Congress slightly over $1 billion to work on this, and we are assembling a meta cohort of COVID survivors and controls to try to understand what's the mechanism of this and what might we do to help those who are affected and maybe to prevent it from people who are just currently being acutely infected. A lot of questions there. One particular input in that is called C4R, the Collaborative Cohort of Cohorts for COVID-19 research. That one alone encompasses more than 50,000 participants to try to figure out predictive factors for disease severity and long-term health impacts, including 14 long-term cohort studies, young adulthood to elderly, following participants for decades. So C4R and IHCC may hold some shared opportunities to work together. Let me conclude, though, by also talking about the issues that relate very centrally to everything that IHCC is trying to do, and that is a global data research commons. The solutions we need to merge data from multiple cohorts, which we have all appreciated, are not at all trivial and require painstaking manual phenotype adjudication and building large consortia. IHCC, along with GA4GH, are potential pathfinders here that can really set the tone of the entire effort to try to optimize this kind of data research sharing. Open science is a core value, but I think we all know there are persisting data stovepipes that we have to address and seek to dismantle while still protecting the privacy and confidentiality of data from individual contributors. So sharing of that kind of aggregated data is a substantial rate limiting step in the development and prevention of prevention and treatment strategies. We have the issue about data non-comparability, coding heterogeneity, difficulties in the storage and transfer of large data sets. And I have to say also regulatory and legal frameworks that are not designed for global sharing can get in the way. And I have to, in this instance, call out, because it's been a considerable source of challenge for NIH, the GDPR, which does not really seem to be well designed for global data sharing for research and continues to require urgent and thoughtful attention. We also have issues about how to share genomic data, not just of human genomes, but also of pathogen genomes, and finding the right balance between protecting the interests of the data producers, who deserve credit for what they have done, but also achieving rapid data access, especially in the face of a global pandemic, is an ongoing issue, as is now being debated for GISAID, which WHO aims to, in the fairly near future, set up a further discussion about how to optimize that, because there are still difficulties in terms of access to data that's being submitted in a way that would really benefit to the maximum level the effort to track new variants that are obviously a source of considerable concern for COVID-19. Well, to wind up, congratulations to IHCC and to all of you for this progress. This has been really phenomenal, and this is one of the most challenging concepts one can imagine, putting this kind of cohorts across the entire world together. But this is emerging as one of the most powerful approaches available to turn the concept of precision health into reality. COVID-19, a global pandemic, makes it more clear than ever how important it is that we have a global approach, not just a domestic approach to precision health. And you, IHCC, are uniquely positioned to play a critical role. It is very gratifying for me to see the seed of an idea presented to heroes back in 2016, now maturing so rapidly as manifested in this remarkable summit meeting. And so I want to express my thanks and my best wishes to all of you as you conclude this working meeting with a very interesting and exciting panel devoted to COVID-19. As the director of NIH, I pledge my continued support for what IHCC is trying to achieve and express thanks and really admiration for all that has been accomplished already. And with that, turn it back to your meeting organizers to carry on to the next panel for this summit meeting. Many thanks.
Welcome to the next to last session of the IHCC. I wanted to take the opportunity to thank the organizers for inviting me to help this session. I really appreciate the opportunity to talk with you and to introduce our great speakers today. So this morning, I'm going to do brief introductions to allow the speakers the majority of their time to actually update us on what they've been doing on COVID-19. Dr. Andre Bernoni is a psychiatrist and associate professor at the Department of Internal Medicine and Psychiatry at the São Paulo Medical School, who's been leading the IHCC Mental Health Working Group and will be sharing results from the ELSA Brazil study. Dr. Cobus Herbst is the principal investigator of the Population Intervention Program and serves as the director of South African Population Research Infrastructure Network, SAPRIN. He'll be discussing the SAPRIN response to COVID-19. Dr. Ricardo Verdugo is an assistant professor at the Human Genetics Program of the University of Chile. He's a director of Chile Genomic project and also leads UGenoma. And he'll be discussing the COVID-19 Host Genetics Cohort Chile, the COVID-19 HG project. Dr. Hendrik Ullem currently serves as the CEO of Statens Serum Institute and is the chief physician at the Department of Clinical Immunology at Ringhospitalet. He'll be discussing how the Danish IHCC cohorts tackled the COVID-19 challenge. Philip Awadala is a director of computational biology and a senior investigator at the Ontario Institute of Cancer Research, professor of population and medical genomics at the University of Toronto, and executive scientific director of the Ontario Health Study. He's the director of the Genome Canadian Data Integration Center and the national scientific director of the Canadian Partnership for Tomorrow's Health, CanPath. He'll be discussing how CanPath approached COVID. And finally, Dr. Michael Gaziano is a preventative cardiologist and internationally recognized chronic disease epidemiologist who's the principal investigator for the Million Veterans Program. He's going to be discussing how MVP included veterans in the COVID-19 research. So I'm going to let our first speaker start, Dr. Bernoni.
So thank you so much for the introduction. Thanks so much for the presentation. It's a pleasure and an honor to be here presenting data from ELSA Brazil. I'm going to talk about these topics. So a little bit of introduction about mental health during the pandemic, some findings from the cross-sectional studies, some findings from ELSA Brazil, our interpretation of the results, and what should we do in the future to move forward regarding this aspect. Next one. So first, I would like to introduce some members of our ELSA Brazil team, especially Professor Ben Senhor and Lotufo, our PIs, and also the students, Daniel Fattori, Paulo Swain, and Pedro Bach, who are helping us. And we also have a grant with Professor Jordan Smoller from Harvard and Professor Sarah Bauermeister from Oxford to research on this topic. As you know, the COVID pandemic started in late 2019 and rapidly spread throughout the world. And with this, we had many disruptions in everyday life, next one, such as travel restrictions, use of masks, quarantines, school closures, and business impact as well. And because of that, we expected a huge impact on mental health. First, cross-sectional initial studies showing variable but high prevalences in terms of anxiety, depression, PTSD, psychological distress and stress, and showing groups that would be especially more affected, such as next one now. You can go next one in the next three ones. So women, younger age, people who are unemployed, and also next one, people with mental disorders as well. Next one. And because of that, we decided to study this matter here in Brazil, in São Paulo, using data from the ELSA Brazil study, which is a study that started a cohort which started in 2008, and therefore we have pre-pandemic data. Next one. So the quarantine and the lockdown started in São Paulo in March 15, and we rapidly arranged a means to do some virtual assessments in ELSA Brazil. So this was the first experience of a full digital assessment, but we rapidly managed to do this with funding from our local foundation, research foundation, and also with funding from IHCC as well. Basically, we aim to evaluate the rate of psychiatric disorders before and after the pandemic, and also to investigate risk factors for developing mental disorders. We already published the main results in psychological medicine. Next one. And we found, maybe surprisingly, but we did not find an increase in the rate of mental disorders before and during the pandemic. We also have to remember, on the other hand, that these rates remained high. So 20 to 25% of people had at least one common mental disorder. When we evaluated the longitudinal assessment towards along 2020, we found also no increase and surprisingly, a trend for decreasing the symptoms of depression, anxiety, and stress during the pandemic. So we found some risk factors associated with more mental disorders, and basically those were, I'm sorry, this is inverted. So risk factors was less than 60 years old, above 60 years old was a risk factor. A protective factor. Being a woman was a risk factor, having non-white ethnicity, absence of college degree, loneliness, concerns about income, increased distress, and low quality of personal relationships were also risk factors for developing a mental disorder during the pandemic. Now we want to tackle more the heterogeneity of the pandemic impact. One study that we are doing is a network analysis. And if you can look very closely, so this is the network analysis before the pandemic. Next one. Here is the last assessment also before the pandemic. And now take a close look and the next one, please. So you can see a change in the pattern of connectivity between symptoms during the pandemic. So the network actually is more connected, and there are theories of mental disorders showing that more connectivity is associated with increased risk for developing mental disorders. The next study that we are preparing is evaluating the trajectories of these people. So I just showed average values. Now I'm showing individual trajectories. We can see that most of patients, or most of participants, fortunately, are presenting very good trajectories, but we can see that there are some participants developing a relatively poor trajectory, and around 5% of the sample actually developing or maintaining a very bad trajectory in terms of mental disorder. So our next step is trying to evaluate that. And now we have this question of what's going to happen in these participants and worldwide as well. So are we going towards the pathway of deterioration of mental disorders or recovery of mental disorders? Next one. This is in line with strategies from the UN Research Roadmap for COVID-19 recovery. This was a series of specialist meetings that happened in the UN, in the World Health Organization, and basically the idea is that the Sustainable Development Goals have been challenged to be accomplished by 2013, and because of the pandemic, of course. But we can try to do some things that instead moving towards the status quo outcome, the business as usual, as they say, we can try to do some transformative changes and develop or achieve a better future or a better development and achieve the Sustainable Development Goals. And the final one, so we have this, we are working with this framework nowadays. So we have the situation before the pandemic. Next one, we can go to worse than before. Next one, we can have the status quo. Next one, we can go towards the pathway of recovery, and many things are going to determine that in our, that's our hypothesis. So next one, basically we can have pathways of mass vaccination or chloroquine as a example of fake treatment. Next one, and the next two ones as well. So we have the pathways regarding developing post-traumatic growth and resilience. of connectivity between societies, of economic recovery, and the other side we have the pathway of rumination, of economic recession, dynamic societies, and now we want to understand which individual and collective factors are going to influence towards one pathway or the other one. Next one. I would like to thank you again the opportunity of presenting this our results. Thank you.
Thank you for the opportunity to share our experience at SAPRIN. SAPRIN is a network of whole population cohorts, each approximately 100,000 individuals. Our COVID-19 related work took place mainly in the 3 rural nodes in the northeastern part of South Africa, here highlighted in blue on the map. The graph shows the number of COVID-19 cases notified in South Africa during the first 3 months of the pandemic, with the timeline of SAPRIN's response superimposed on it. Within 10 days after the WHO declaration of a pandemic, we had agreed on how to amend the existing surveillance protocol, switching from 3 household contacts per year, one of which was a physical visit, to exclusively telephonic interviews. This was done to comply with the strict lockdown conditions imposed in South Africa and to prevent exposure risk to our field staff and surveillance community. We also added a telephonic household impact questionnaire and symptom screening of all resident household members. Suspect COVID-19 cases were either referred to local health services for confirmation and testing or to mobile testing points close to the household dwelling. Taking advantage of expedited ethical approval mechanisms by IRBs in South Africa, the amended protocols were all approved within the 2nd month following the pandemic declaration. The new protocol was in place before there were any community transmission cases notified in our surveillance population, and the protocol is published in Wellcome Open Research. One component of the amended surveillance protocol was a random sample of 750 households from each node that were telephonically contacted every 2 weeks to get a finer temporal resolution than the 15-week contact cycle for the rest of the approximately 250,000 households and 25,000 households in each node. The figure at the top shows the progression of the 2 weekly surveillance over the course of the first wave of the infection. The small graph shows some of the attributes measured longitudinally in the same households over the first 7 months of the pandemic. An ongoing analysis of the data allowed us to present at several ministerial seminars which influenced national policy, and as a result, SAPRIN is now a founding member of a national data observatory with direct reporting lines to national ministerial level. The analysis of the data is published and in that reference on the slide. This somewhat complicated graph shows the official notified COVID cases in the surveillance population in the gray bars, with a number of individuals self-reporting of being diagnosed with COVID-19, blue bars, or testing positive through our COVID PCR screening, orange bars, across 2-week intervals spanning the first wave of infections at one of the SAPRIN nodes. The yellow line shows the limitation of our surveillance approach. In any 2-week interval, at best, we could reach around 8% of the total surveillance population of 100,000, allowing us to directly identify only a fraction of the actual COVID-19 cases in the population at large. A more sustained measures of SARS-CoV-2 exposure, such as seropositivity, will allow us to have a better handle on the epidemic in our population, of course complicated if and when vaccines are rolled out in the study population. Although we are now routinely collecting dried blood spots from the surveillance population, we have been unsuccessful in obtaining funding to test these for SARS-CoV-2 antibodies, an example of the inability of LMIC cohorts to convert samples into data amenable to scientific study. At the moment, we are focusing on assessing vaccine hesitancy and reporting changes on an ongoing basis to local and national health services. This is part of a body of work that relies on our ability to link our population database to health service records to look at the impact of the pandemic in general on healthcare access. We're also part of a study on 10 low middle income population cohorts to assess the impact of COVID-19 on all-cause mortality. And you can read more about this work in the listed citations. Thank you very much.
Hello, everyone. Thank you for the opportunity to be here and present to you the results of a new cohort to study COVID-19 in Chile. Next, please. Well, I will walk you through the objectives and design, challenges, status, and perspectives of the study. Next, please. So in Chile, as you may know, we basically have only one dimension, north to south. But along that dimension, we are a heterogeneous population made of the admixture of Europeans and several indigenous groups. And I have listed here along the axis the different groups that were the major contributors to the admixture process that made today's population in Chile. And to study the genetic diversity in our population, the Chile Genomic project previously sampled 3,000 volunteers along the country from Arica to the extreme south. And of those, we genotyped 414 with Axiom LAT1 microarrays, which has about 600,000 effective SNPs. Using this information, we estimated polygenic risk scores on the population to determine what would be the predicted risk for susceptibility of COVID-19 using the results of a 23andMe study that was recently published with over 1 million participants, of which 15,000 were COVID patients. And they detected 2 independent loci in chromosome 9 and 3. And we selected 1 SNP per locus and estimated a polygenic risk score in the 1,000 genomes reference populations of African, European, East Asian ancestry, and also in Aymara and Mapuche, and in different regions of Chile. And what we found was that Europeans and Asians will have a predicted higher score of susceptibility than the Aymara and Mapuche, and we Chileans will be somewhere in the middle between these two groups. We also did the same for a different PRS with considering 8 SNPs taken from the Genomic study that was based on 2,244 critically ill COVID patients. And what we found was the opposite. Now, the Aymara and Mapuche indigenous populations are predicted to have the highest risk for the severe forms of COVID-19 as compared to the East Asians, Europeans, and Africans. And we, again, are somewhere in the middle, the majority of the admixed population. So this suggests that risk may be associated with ancestry in an admixed population in Chile. And therefore, it is important to study whether in the genomes of these admixed individuals, there are other, first to confirm that there is a differential risk, and also whether there are other elements of risk that are not included in those published studies. Next, please. So we took the opportunity given by a recent call last year to study COVID-19 that gave $100,000 to study to several studies. So we were selected to study the effects of the host genetics, where we are interested in identifying these genetic loci that will differentiate the severity of the disease and whether that severity is associated with ancestry. So we started with a recruitment in a pilot in a hospital here in Santiago in September and October 2020. And then in November, we opened a call to the whole population by several media. And over 3,000 people contacted us through a contact form in Google, and then we returned calls. And people were either appointed to come to a recruitment center that are listed on the left map, or were visited to their home. So the objective was to recruit 4,000 people with surveys and genotyping with a GSA microarray to perform a GWAS. We used a survey that was based on the Wellcome Trust and IHCC survey that considers demography, ancestry, socioeconomic, comorbidities, 26 symptoms of COVID-19, medical care, and some sequelae after COVID-19. All of this was implemented in REDCap and translated from the original templates and also adapted to the local reality. And all the sample processing was using FAIR guidelines, and this was only possible thanks to our institutional biobank that it really taught us how to do this. This was the first time that we worked with SOPs and numerous record forms to be able to trace all processing steps of every sample across the different recruitment sites across the country. Now, the challenges are basically engaging the clinicians that are overworked. And so that's why most of our volunteers came directly and not through hospitals. We had a couple of hospitals that participated though. IRBs, one per hospital, made everything very slow. And now we need to harmonize data across different projects with which we are collaborating, and we are seeking funding for follow-ups. The current status, we completed 2,000 volunteers. So this is 50% of what was planning in the beginning, and that's because it was very much slower than what was expected because of the reasons that I just mentioned. But the good news is that since we were several projects that started at the same time to study COVID in Chile, we created a consortium together that added 1,500 more participants to the study, all of which will be genotyped either through our study or other studies. And all DNAs were already submitted recently. Actually, most of them submitted last week, and next week we will submit a second batch to either the Broad Institute or FIM, which are our partners for that part. And you can see on the map that we have different levels of severity from asymptomatic to lethal, although the most common it's in Spanish level, it's mild. Mild cases are the most common. And we were much successful recruiting women than men. And I want to acknowledge the different PIs. This is a collaborative effort with participation from several institutions, as I mentioned, and also our international collaborators are the Broad Institute and FIM that made this possible, and our sponsor, the National Agency of Science and Research. Thank you very much.
Hello, everyone. Thanks for the opportunity to talk here. Could I have the first slide, please? So my name is Sentinel. I'm the chair and the CEO of the Statens Serum Institute, which is an institution that monitors the Danish COVID epidemic. But also here we host the Danish National Biobank. So I'm going to talk a little about the infrastructure that we have where we can do these COVID-19 studies. Next slide. So the biobank is a very large biobank. We have 12 million samples in store, and that is like the backbone of what we can do of COVID-19 studies, genetic and other biomarkers, and including all kinds of gear, including genetic analyzers for large-scale genetics. The important part is that in Denmark, we have these wonderful health registers with the basic central personal registration number. We can track all individuals, so we can kind of follow both COVID-19 status, but also other kinds of healthcare status. And in the National Biobank, we also have the National Biobank Register, where a large number of Danish cohorts are represented, including the IHCC cohorts, and also potential future cohorts. I'm going to talk about these a little later. So here at Statens Serum Institute, as mentioned, we monitor the epidemic, and that includes a large number of data sources. We have the National Microbiology Database, Vaccination Register. We have information from nursing homes, from occupation and workplace, from the civil registration system, and from the National Patient Register. And importantly, we actually sequence each individual virus sample, so we can do these kind of very advanced post pathogen interaction studies genetically. So already in the Danish National Biobank, we are running a large number of COVID-19 studies. I'll not go into details with these. You can just see these. And then I'm going to talk about the IHCC cohorts. The first one is the national birth cohort, which was included from the mid-90s to 2003. It's around 100,000 children, and they've been followed ever since with a number of follow-ups. And at the end of the follow-up here, you can see there are some important COVID-19 mental health follow-up. And we are, of course, looking forward to see the interaction between genetic results and the mental health in these follow-ups. Another important, although not yet IHCC cohort, but I think it should be, I was the one initiating this cohort, the Copenhagen Hospital Biobank, where we have 430,000 patients, and approximately half of them has been Illumina GSA chip typed. And as mentioned, we have this very extensive phenotypic data. And for the ones of those that then have encountered COVID-19, they can, of course, be included in the COVID-19 studies I'm going to describe for you. And another very large population study, which I was also involved in initiating, that's the Danish blood donor study, where we, for 10 years, have prospectively followed 135,000 blood donors with surveys, blood samples, and all these individuals have been genotyped as the other previous cohort. So right now we are combining all these data in a large study describing the epidemiology and genetics of COVID-19, and the objective is to acquire knowledge of the COVID-19 disease pathogenesis and trajectories. And of course, we will like to focus a lot on the host genetics, environmental factors, comorbidities, viral variants, and strain and microbiome. And the idea is that we will want to develop a precision medicine approach to COVID-19. And in this study, we will draw data from all the cohorts I described and from the biobank. And right these days, we are sending out messages to 26,000 participants on top of the ones that are already included that have been infected with COVID-19. And we do have their samples in-house, so we just need their consent, and then they are kind of good to go for the project. So the population we're including, it's disadvantage, it's majority European origin, but as you can see on the top to the right, we also have other ethnicities. We have developed an algorithm to do high-resolution HLA typing from imputed genotypes. And then, as mentioned, we can link these very rich genetic data with very rich phenotypic data, and we can combine host and viral sequence data. And with that, I want to finish, and thank you for listening.
I'm at the University of Toronto and the Ontario Institute for Cancer Research. So I'm going to be giving an overview of the Canadian Partnership for Tomorrow's Health cohorts activities in the cohort and the COVID research space. Next slide, please. But first thing I'm going to describe is what CanPath is. So CanPath is our national population cohort, which has recruited and consented over 300,000 Canadians. It's a partnership. We have 10 provinces in Canada, so it is a partnership across those provinces. Those 330,000 participants are largely spread out, mostly evenly across those provinces, with the exception of Ontario, which has a larger population. Of course, we're more representative in that population. And the cohort has captured lots of information since 2009, in fact. So it's a longitudinal cohort. We've been following these participants over years, and naturally we capture many of the same things that many population cohorts across the world are collecting. So as mentioned, it's a partnership. We have 10 paying systems, or 10 payers in Canada. So our health system is such that it requires a partnership, particularly with regard to sharing data across these provinces. Next slide, please. And so what I wanted to focus on is what our COVID program, specifically called the Support Canada program. This program launched in March of last year when Canada effectively went into its first lockdown. And it's a program where we're capturing from over 100,000 participants within the CanPath activity data specifically with respect to COVID-19. You'll see some of that information in a moment. We're also capturing serological information as well as genetic information to assess host and viral sequence information, host pathogen interactions. One of the things I wanted to quickly highlight is that CanPath itself is fairly representative of the national ethnicity profile, if you like, of Canada. We're largely a white cohort. 75% of the population in Canada is white, and so CanPath reflects that. But because of our size, we're able to, if you like, recruit and engage in some of these understudied or underrepresented populations. So for some of our serological data that you're going to be hearing about in a moment, we've been targeting some of these populations that are described here on the right. Newcomers to Canada, self-declared Indigenous, long-term care residents, and people who are living in high prevalence regions. So some of the things that we're asking in our baseline questionnaires from our first 100,000, this started in March of last year, and we continually are updating this information. We're in our third wave of the pandemic in Canada presently, so we're updating this through each one of these waves. We're asking about participants, about their test results, what symptoms have experienced, hospitalization, travel history pre-pandemic, impact on job status, and of course mental health questions. And so we're capturing information about how behavior, in terms of behavioral responses as well. So one highlight I'm going to identify here is sex and gender differences in the cohort. Women are more likely, for example, to follow guidelines with regard to wearing masks, staying home, and so on. We're seeing a lot of sex and gender as well as ethnicity impact on the data. For example, within Canada, we're seeing some substantial racial inequities. 2.1 times ethnic minorities are 2.1 times more likely to be infected with COVID. That speaks to a number of different factors, socio-demographic and work histories. And so again, this speaks to that as well. Socioeconomic factors are playing a large part and are tied to ethnicities with regard to COVID-19 infection. One thing I will just pause and say is all of this data is now available through our data portal, and we're open to partnership with other programs across the globe who are capturing similar types of information for their studies. Serious gender gaps in the data. We're seeing nationally that women are certainly overrepresented in certain industries, and which are of course being impacted more severely with regard to COVID-19 diagnoses as well. And of course, something that was mentioned by Francis Collins during his presentation is long-term effects. So we're following, because this is a longitudinal study, we're tracking over time people who are exhibiting symptoms of long-term effects with regard to COVID-19 as well. So we're knee-deep in a serological study. We're capturing data from our target was, and funded by CIHR and the Public Health Agency of Canada, was to target 20,000 individuals, particularly from people within certain job classifications, people who are hard hit with regard to regions or hotspots, if you like, as well as some of the groups that I mentioned in my previous slide. And we're also finally in a place where we're now seeing that in CanPath, actually, 65% of the cohort is already vaccinated, receiving at least one single dose. Canada is in an interesting sort of experiment where we're seeing close to 60% vaccination with single dose, and yet only 4% vaccinated with double dose, or 2 doses of Pfizer, AstraZeneca, and Moderna. Now, that has a lot to do with our limitation. We don't actually have our own vaccination manufacturing in Canada, so we're in an interesting experiment where we spread out almost 16 weeks between first and second dose. So time will tell whether that will be effective. So some of what we're trying to capture with regard to our serology is, what are the signatures with regard to long-term antibodies? How long lived are those antibodies, post-infection? and also with regard to what I was just mentioning with regard to vaccine efficacy. We wanted to target 20,000 people. We asked questions, asked people, 30,000 people, and we're seeing actually 90% participation rates, which is pretty astonishing. If you're ever doing anything in population cohorts work, as many people here are, we're pretty impressed with our response rates. Next slide, please. And so some of our first analyses are coming back now. As I mentioned, 68% of CanPath has already received 1 dose of Pfizer, Moderna, and AstraZeneca. That's being reflected in the serological tests. There's an 8-week lag between when somebody's finally first invited to provide a blood sample to when we actually get information on 3 antibodies, and you're seeing the pictures of how those 3 antibodies correlate. 2 of these antibodies are reflective of the vaccines that are being administered in Canada, and 1 is more specific to viral immunity, and so we can discriminate immunity, if you like, associated with vaccine versus viral. And so we're actually seeing some serious lags in the viral immunity, whereas we're starting to see it certainly increasing with vaccine immunity with regard to serology. So that's data that's coming off the press. This project will be done by June, and we'll have data for 30,000 people. As I mentioned, this is my last couple of slides. This is the portal where you can get access to all the data that I just described. So this is the CanPath portal. And my last slide is to thank the leadership team. And actually, there's one more slide as well. So these are our scientific leaders, and of course, our National Coordinating Center at Toronto, who are spearheading this activity. So thank you very much.
So it's my pleasure to share with you MVP's perspectives on its response to COVID in specifically, and in general, the utility of live cohorts for rapid response and the kind of coordination between cohorts that IHCC presents. So this is a snapshot of where we are with the Million Veteran Program, with 838,000 individuals recruited after 18.5 million mailings to 6 million people. When the pandemic started, we had 828,000, and we recruited 10,000 over 14 months, far off from our about 8,000 a month recruitment prior to the pandemic. I will point out that this is about 30% underrepresented minority-based cohort. Next slide. This is a heat map of where our MVPers reside, and not surprisingly, it coincides with the hot zones for COVID, making this a wonderful cohort to study nationally and even look at geographic differences. Next slide. And I'll just point out a couple of landmarks. Of March 16th, we closed sites and moved to online recruitment. May 5th, we released our online questionnaire, and by June 5th, we had 6 scientific working groups up and running. Recruitment shifted to online. I'll share a little bit of information about that. We have surveys out not only on COVID, but also on vaccination and on long-haul COVID. They'll be released in the late summer. We've explored in-home biospecimen collection, and then scientific collaboration within MVP working groups and with outside collaboration. So online, we had to shift all of our effort from the sites to calling participants to get them to sign in, consent, and then we disabled the scheduling portal and began moving toward remote blood sample collection. We coordinate globally the development of our COVID questionnaire, and we have 250,000 responses from a mail out and the online portal, and which were integrated with our existing cohorts and cohort questionnaires. We did piloting of our mental health cohort, that's the mind, and then we've got development of our vaccine and long haul COVID questionnaire, again in collaboration with other cohorts. Next slide. Now, we needed a place to share the data. So Cypher is our phenomics portal used by MVP investigators to share. And we've got about 4,000 phenotypes, algorithms, codes, meds, labs. And this portal was supposed to go live for all of VA investigators to use beyond MVP later this year. But we raced it to release on April 16th so that we could release COVID data to all VA investigators. This is the landscape of COVID within the VA. About 1 in 6 of the 1.2 million people tested, or 224,000 people have tested positive within VA. Some tested positive outside. In MVP, it's 33,000. And we have genotypic data with imputation on 26,000 available. And those are the 3 tranches of data We can study COVID in the VA at large among 200,000 individuals. We can study it with questionnaires on 33,000 and with genetic data on 26,000. And as an example of harmonization, we took the WHO severity scale and developed a 4-point scale of not hospitalized, but with COVID, hospitalized with some severe indications such as ICU or intubation and death. And these data are, again, available on the CIPHER website and are used by many investigators throughout the VA. Then we tested online. I'm going to briefly mention basically one. We had been testing saliva DNA collection, and while the qualities of the DNA is good, the compliance with being able to do it properly on a one-shot basis was about between 50 and 60%. So we tested a capillary device called TASO, a blood spot called Mitra, and mobile phlebotomy. And we settled on the TASO device, which is a capillary device that you attach to your arm. And it has a little puncture device that retracts and collects capillary blood. We had, by phone, 53% consent rate and 94% devices returned. The DNA quality is excellent. The plasma sent overnight, not on wet ice, is good for the COVID assays. We're testing metabolomics and proteomics on that plasma as well. And the new device, TASO Plus, coming out this summer, will collect as much as 700 microliters of blood through this mechanism. So moving very quickly to finish up my talk about this on the science, the priorities were speed, collaboration across existing people who knew how to work with the data, and in collaboration with outside entities such as HDI. So 6 working groups were created: disease mechanism, druggable genome, genomics and PRS, pharmacogenomics, PHWAS, GWAS, and 2 core, just the epidemiology of COVID in the VA and MVP, and then cohort management and methodologies. And you can see that there's many papers published, in press, submitted, ready for submission in each of these domains. And I'm going to just finish with one example. This allowed us rapidly to use a 2-sample Mendelian randomization technique to search for potential existing drugs that could help with COVID. This was under the direction of JP Casas, our executive director for MVP, and Adam Butterworth at Cambridge. And through this 2-sample Mendelian randomization, 1,300 proteins that had variant instruments were cross-referenced with variants that predicted outcome. And then through a colocalization process, 2 very interesting receptors, IFNAR2 and IL-10 beta, turned out to be direct hits, suggesting that interferon may be a wonderful drug target. And this compresses the ability to look at the development of drugs, and there are early trials of this in early COVID that are in development. So it demonstrates the ability to rapidly respond in this one way. And if any other publications, like, we can provide that information to anyone who's interested. I just want to point out that existing organic cohorts are critical for being able to respond rapidly to a crisis like this. And collaboration, like in IHCC, which we use for harmonization of our instruments, I think is essential. And I always end by thanking our most valuable partners, which is our veterans. Thank you very much.
Terrific. Those were outstanding presentations. And I want to thank our presenters on really very thoughtful and inspiring work that's been done in very short order. And it's been exciting to see the evolution from conceptualization of questions in March to actually publications now, and how far we've actually come. Regrettably, we don't have time to take any questions. However, the panelists will be available in the chat, and so hopefully you'll be able to ask questions then. I would like to take a moment to introduce our panel who's going to come and speak with us today. We have the opportunity to speak with several very established investigators. Our first is Ms. Mader from the Illumina. She's the Director of Medical Affairs for Global Health and Infectious Disease at Illumina, where she leads efforts to expand access to genomics for pathogen surveillance and infectious diseases, to foster international collaborations, and to drive standards and evidence generation for pathogen genomics. She recently led the deployment of new assays and tools to drive diagnosis and molecular epidemiology in the SARS-CoV-2 pandemic response. Next, I want to introduce Dr. Amagari. He's the director of the AMR Clinical Reference Lab and director of the Disease Control and Prevention Center at the National Center for Global Health and Medicine in Japan. He's been acting as director of Disease Control and Prevention Center since 2012, and also serves as director of the AMR Clinical Reference Lab. which is commissioned by the Ministry of Health, Labor, and Welfare. Finally, I want to welcome Mr. Leo Wolanski from the Rockefeller Foundation, where he does data analytics and is the product lead for its Health Initiatives Precision Public Health and COVID-19 response programs. He focuses on identifying opportunities to leverage digital technologies that would strengthen health systems and improve health outcomes. Throughout the pandemic, he has managed programs across diagnostics, digital innovations, community health, and public health intelligence in multiple continents and globally. And now he's focusing on implementing the Rockefeller Foundation's recently announced plan to build a pandemic prevention institute. So I welcome all of our panelists. Thank you for joining us today. We appreciate your time. And I'm going to take the moderator's prerogative of asking the first question, which is, how do you think that IHCC cohorts can be leveraged best? We're not, for the most part, thinking about ourselves as public health types of ways to identify pandemics, but how could IHCC cohorts be used to better understand and learn from pandemics? And in the interest of sort of making sure we all have a chance to answer, I'm going to go with Vanessa first, and then we'll sort of go around the Zoom room to make sure everybody has a chance to talk.
Sure. Thank you, Kelly. I really think these cohorts offer such a unique opportunity in pandemic response, whether it's for this one or future pandemics, because of the rich metadata that we have on these patients and participants. We can learn so much with these new pathogens where we really don't understand what's making people sick. Why do some people get seriously ill and others do not? Being able to mine some of this data and have longitudinal access to look in sometimes in real time and watch responses and watch who falls ill and who doesn't. It's a way of learning that we've never had before with previous pandemics. Even if you look at Zika virus or H1N1, we have empowered the community to develop vaccines, to understand best therapies by looking at host factors. And I think having these cohorts is absolutely essential, and being able to mine that data and pick out nuances of different ethnicities and risk factors is really, I think, unparalleled. And so having these cohorts, while maybe intended for other uses, because of the rapid accessibility of some of the data that you don't necessarily have to go prospectively collect to start understanding, is incredibly useful.
Terrific. Thank you. Dr. Amagari, would you like to respond as well?
Thank you for the time for giving me a time for the talk. And honestly speaking, I'm not familiar with this network. However, I can definitely say that the importance of the clinical information and also the information about genetics data from the human being and also the pathogen as well. So the most difficult part of the response to the this kind of pandemic and the emerging infectious diseases is a lack of data. Especially because as we noticed in the very beginning, the dissemination of the COVID-19 had sort of the laterality. In some part of the world, the epidemic was quite bad, but in other part of the world, it was not so bad. So, in the part of the world where the infection was not so endemic, it was quite difficult to obtain information, of the clinical information, and also about the information about COVID-19. But in that case, this kind of cohort will really help to obtain data, even though we are not yet in the the epidemic or pandemic. So that is my contribution.
Thank you.
Thank you.
Leo, you have a lot of experience with looking at pandemics and data. What are your thoughts?
One of the key things about public health data is that it's often developed in silos. And so you get kind of one data stream independent and not linked to any others. And so the implications in terms of interpretation for actual public health decision makers is quite limited. And so what they have to do is look at the science, and this is normally in the forms of papers that inform them around what the implications are. And one of the challenges for public health decision makers, especially in a health emergency, is that there's a lot of information to comb through. And so one of the key things that these longitudinal population studies can offer is if they can align some of the data streams in terms of what kinds of data is being collected for public health, and also collecting very similar or ideally in the same standard, the data through their studies, they can very rapidly generate scientific insight for public health decision making that would be totally impossible to acquire from just the public health system alone. One particular thing that this pandemic has catalyzed is a proliferation of a lot of new types of data and sensors for public health. Probably a few years ago, no one would have known what a nasopharyngeal swab was or what a variant was. And furthermore, many people are now willing to give their data for all sorts of things, to stand and wait in a wait list for maybe getting a vaccine appointment, to take surveys every few days on whether they're healthy or sick and what symptoms they're getting, or wear a device on their ring or on their finger or on their wrist, and share that data in real time with public health decision makers, with governments, and with scientists. One opportunity we have is to incorporate those types of data, ideally in a pretty similar format, into these longitudinal population studies, because there will be a lot of this collection ongoing for some time, or one might think. And so that's a good opportunity for us here, is to generate these insights for the long haul so that we're ready for the next time.
Terrific. Those are all great suggestions. I'd like to invite our attendees to please put your comments in the chat so we can help with moderating those questions. In the meantime, I'm going to ask a question about how do you think cohorts might be utilized to look at the impact of long COVID? So we've talked a little bit about that in some of the presentations. Francis certainly mentioned that in his talk. It's affecting a large proportion of people, regardless of whether you had severe COVID and were hospitalized or whether you had mild COVID. And it doesn't necessarily seem to be related to some of the things we traditionally think about in terms of comorbidities or age. It seems to be affecting huge numbers of people. How can we use our cohorts to potentially think about that? And maybe in the interest of sharing the wealth, Dr. Amagari, I'll go with you first, and then we'll sort of work.
Thank you. In our country as well, the long COVID is a huge issue, and the most difficult part of the understanding of the long COVID is how I can say. Probably this is this is a syndrome, and it contains multiple type of diseases with quite different etiologies. And the most biggest obstacle is to find out what is the mechanism behind that. So to do that, we need multiple ways of the analysis, including genetic analysis and also phenotypic analysis, including the proteomics analysis as well. And also we might want to look into the how I can say social or mental side of the patient, which may explain some part of the long COVID. So what I mean is this is a multidisciplinary issue. I mean, the long COVID has multiple etiologies probably, and to solve all the issues, we do need a various type of the different types of data from the natural science, between natural science and the social science. So to do that, we do need this kind of cohort, and we do need different types of the the data to find out. So so we just in one word, it's really helpful to have this kind of the cohorts. Yeah, thank you.
Leo, would you like to tackle that?
I think Dr. Omagari covered the critical things, which are how do you understand the drivers of long COVID, and then ultimately start to understand its impact. On the first matter, on understanding the drivers, I mean, similarly, you could be trying to sequence viral pathogens to understand whether there are certain mutations that are driving some of these differences that we're seeing, as we really don't understand why some people develop long COVID and others do not. And so what are some of the underexplored data inputs on these longitudinal population studies are ones that we should consider adding. On the impact side, we might not know fully what the drivers are, but at least these longitudinal studies can help us understand what some of the symptoms are consistently and help us better measure the prevalence of long COVID, which, unlike much of the rest of this current pandemic, folks are much less likely to go get medical care for this. And so there's actually a pretty limited understanding of what the health and economic impacts right now are of long COVID. And that could be another really important thing to help us learn in the near term.
Absolutely. Thank you. Vanessa?
Yeah, I think, I think as the others nicely stated, this is a complex issue, and I think large data sets can only help us better understand. I do think it's very likely to be a syndrome with many causes. I think the notion of it having potential pathogen factors is really intriguing. We certainly think of host factors and predisposition to autoimmune diseases as a major issue, but I think taking a multi-omic approach to really doing a full investigation on these patients can only help to shed light on something that is a total mystery right now. And until we understand the root cause, I think we're going to have a really difficult time managing it. And what I think these large cohorts help to do is to wash out some of the noise, to help us get to the essence of the true causes behind syndromes and issues such as long COVID. So I think, again, the more data we have across a wide diversity of patients, the more we're going to be able to uncover. But really being able to do these multidisciplinary, multi-omic approaches, I think is going to be our best bet, because it's quite likely that some of the cause is not where we expect it to be.
I think you're right. We're going to learn a lot, just like we've learned about acute COVID. I think we're going to learn a lot in the coming year about long COVID. Dr. Ginsberg has put a question in the chat. For the next pandemic, what should cohorts be doing to prepare such that they can provide sentinel signals to public health officials? And how do we make sure that the data is made available to allow for a robust response? It was really exciting to see a number of our presenters show that their data is now publicly available. And I think that that's really terrific, allowing lots of investigators to be able to analyze it. I'm going to open up to all three of you. What do you think our cohorts should be doing to prepare for the next pandemic to be able to alert our public health officials?
I can start there and give a few thoughts, and then share with my colleagues here. So, I think the accessibility of some of these cohorts is key. The biobanks are great, but having longitudinal access is incredibly important. So, having access to patients and saying when there is a certain set of symptoms, be it respiratory or others, and eventually in a kind of forward-thinking world, we could send sampling kits to some of these participants at home for them to sample themselves and send back in, being able to collect those specimens and not just interrogate for pathogens, but really understand who they're coming from and what their predisposing factors and comorbidities are. I think that can really add a richness to an early warning type of system, because right now so many things percolate in the background. We saw what happened in the US with COVID and how long it truly took us to understand what was happening. And part of it is just accessibility to patients and to their information. And so I see that as one key way that we can help kind of accelerate our position for the next pandemic.
Terrific, thank you. Leo or Dr. Amagari?
I can provide one particular thought, which is, there's been such inefficiency in the way that we've had to monitor the prevalence of COVID-19. If using essentially exclusively convenience samples, diagnostically generated data on diagnostics, and then trying to leverage this data that is not collected in any sort of representative manner to measure prevalence comes with a lot of risks in terms of bias in how we understand the impact and spread of a virus. And so if we do want these longitudinal studies to become sentinel indicators, they will then need to become a bit more representative of the populations that they seek to represent. And so one idea would be, focusing in on recruiting in areas that are currently underrepresented. And if that means creating almost, you might consider different sub-cohorts within a cohort study, in which perhaps in one sub-cohort, the burden for data sharing is more limited. Perhaps you don't collect full genomes for every individual that's participating, but you're able to get them to participate in a lighter touch manner to make sure that we are actually including more of the population. Those are some of the ideas that I think would be important to consider, in addition to adding some of these new public health sensors like wearables into the studies now.
Diversity is clearly an issue, and making sure that we have the appropriate populations represented, I think, is something we've heard both from our investigators as well as clearly from our public health officials. Dr. Amagari, do you have any comments on this?
Tough questions for me. The thing is, I may not to the point of this question, but I'd like to answer the first question. In our experience in Japan, we found that it's really helpful to have some type of the Sentinel surveillance system to find out the case of the infectious diseases with unknown cause. Unknown pathogen. For example, the first case of the COVID-19 in Japan was found through those kinds of network. Under the infectious diseases law, the government of Japan introduced this surveillance system for the acute respiratory diseases or acute feverish diseases with unknown etiology. And the first case was actually found under that umbrella of that kind of surveillance. So what I mean is, one idea I'm personally thinking about is having a sort of the the surveillance system, cohort system to register the infectious diseases, especially acute respiratory, probably acute respiratory infectious diseases with unknown etiology. So well, it might be the first case of the novel, avian influenza, or could be another type of the, the coronavirus. But anyway, the most, looking in the past, the most of the pandemic came through the, acute respiratory infections. So having a special cohort, Sentinel surveillance system, or in other words, cohort, cohort for the, this kind of acute respiratory infection may help to find out, to detect the first case, and also the season thereafter. So
excellent.
So, yeah.
No, it's terrific. Dr. Gaziano has asked about the biology of disease can be studied within existing cohorts, but how do we make sure that we invest in an understanding to be able to translate this into the general population? So people who tend to volunteer for studies in general are different than the general population. How do we make sure that what we learn from these studies are representative and can be applied more nationally or internationally?
I can take a stab here. I certainly provide one thought. I think it's a really important point, and we kind of don't know what we're missing. We don't know what we don't know. But I think what we can try to do is understand the gaps and where is that selection bias when you're looking at some of these cohorts, and say, what are we truly missing, and be very proactive about going out and interrogating those populations that are being missed. I think it just probably has to be very intentional, and we have to be mindful that this isn't representative of the general populace. And as we work to broaden these study cohorts, at the same time, when responding to public health events like this, I think in near real time, we do need to be mindful of what exactly it is that we're looking at, map that landscape, and say, okay, where where are the dark spots and how can we go fill those in quickly?
Any other thoughts?
I think, obviously these cohorts are doing a lot, and it's important to understand that perhaps not not everything can be studied within the same cohort study. Nevertheless, perhaps if we do want to be able to translate at least some of this information to population estimates, we might need to treat recruitment for these studies more like a political poll than a scientific research study in which we're actually trying to make sure that we collect or include a certain number of folks from a variety of different population groups to actually make it representative of the broader populace from the outset. Otherwise, it's no statistical method can undo bias in the underlying data if you don't collect data from the entirety of the population.
That's a great point. One of the other things that I think is important and something that has been thought about in the IHCC is data harmonization. And how do you take a data element that's collected in a continent and potentially be relevant on another continent or another country? And given your expertise of thinking about this globally, what would you have as advice for actionable ways to collect data sort of moving forward for IHCC leaders?
I'll go first since Vanessa's kindly gone first every time. I think every public health system and the way that data is collected globally is, there are significant differences that make this challenging. I think in terms of, I know that there is a data standardization working group, which is largely focused on addressing this exact issue, working through and in concert with global standard setting bodies is likely what needs to happen, as IHCC likely doesn't want to set those standards itself. And I saw in a number of presentations leveraging standards on COVID severity and diagnoses from the WHO. That is going to be a really important function that the IHCC needs to play, incorporating those standards into its studies.
Absolutely. Any comments from our other panelists?
No, I think Leo nailed it. So, we're going to have to, again, just kind of proactively go out and start to look more broadly at where can we drive efficiency in the cohorts that are being recruited for various studies, and say how can they be leveraged for multiple purposes, and to drive that diversity in recruitment to better serve kind of these broader public health needs.
Absolutely. Well, I know we're at the end of the hour. I want to thank our very esteemed panelists for their generous donation of time. This has been a great conversation. I want to thank our attendees who've been able to send in some great questions, and really appreciate the opportunity to talk with all of you today. So thank you for your great work that you're all doing on behalf of many different cohorts. And I'm going to turn it over to Dr. Ginsberg, who's going to take us home for the rest of the session. So thank you, everyone.
Hello, everybody. That was great, Kelly and panel. The entire 3 days has been incredibly impressive and inspiring. This being our fourth IHCC Summit. I'd like to thank all of our excellent speakers and panelists and moderators and session chairs, and also for those of you that are still here after 3 days to stick with us for the entirety of the meeting. I did jot down a few notes I wanted to share with you, because I think we've covered an incredible arc of information and progress over the last 3 days. On day one, which was Tuesday, which seems maybe like a week ago, we heard from Corey Bargman from the Chan Zuckerberg Initiative, who told us about CZI's strategic pillars. And my observation was these are incredibly well aligned with the IHCC's, them being partnership and collaboration, the ability to accelerate science, the ability to change the culture of science to be more open, to meet the challenges of diversity and focus on underrepresented populations. I think those are all in very much lockstep with what the IHCC is trying to do. On Tuesday, we welcome 7 new members of our steering committee from South Africa, from Africa, South America, the Middle East, and from Asia. And we also heard about progress in all of our cross-cohort pilot programs. And there was an open invitation to new cohorts to join these programs, and we'll provide information to all of you to ensure that you have ample opportunities to be part of the science. We heard about the diverse ways in which the IHCC cohorts are examining aspects of COVID-19. We heard that on Tuesday. We heard it again today. And we will make sure that all of our members see the progress that has taken place so that we have updates from the funded scientific programs that are underway. We also heard about the Davos Alzheimer's Collaborative and the opportunity for us to participate with that group, in addition to the progress that's been made on establishing the IHCC Global Cohorts Atlas that would allow for greater discovery of all the work that all of you are doing. And we heard from our newly named Policy and Systems Working Group about the groundwork that they're laying for enabling policies that will allow IHCC science and data access to take place. Yesterday, the second day of our meeting, Mary Da Silva kicked us off from the Wellcome Trust, talking about the disruptive opportunity for longitudinal population studies, focusing on cohort interoperability and harmonization as part of that disruptive vision. We also heard that we completed and launched a strategic plan, the vision, the mission, and now it's really time to implement our strategic plan with a focus on efficiency and also a focus on being focused. We know from what we've seen over the last year or two that we need to diversify our IHCC leadership. Hopefully our new scientific steering committee membership is a good start. Yesterday, we heard from 4 working groups about where they want to go for the future. So our data and infrastructure working group has put in play the goals of increasing fairness, FAIR, of the IHCC cohorts, the harmonization of our data, the cohort metadata in particular, and then hopefully over the next year or so to complete an expansion of the cohorts atlas to allow for greater inclusivity, discoverability, and advancing the science. And we also heard about the challenge of keeping the atlas current. The Science Strategy and Cohort Enhancement Working Group espoused the goals to expand the science to more and diverse cohorts, to coalesce around funding opportunities, and in particular to engage industry partners in our work. The Policies and Systems Working Group indicated their work to put in policies that will actually be enabling for IHCC science, but also to take a role in developing and leading with other global organizations in policy development, and to create a toolkit that can be used for disseminating policy materials to the global community. And then finally, we heard about a new working group that is now called the Training, Sharing, and Capacity Development Working Group that has had lots of ideas in its very short time with us, but talked about workshops, seminars, symposia, cohort curricula, scientific exchange, and young investigator opportunities to be the cohort leaders of tomorrow. So that is a new dimension to the IHCC that we really are looking forward to. And yesterday, we also had a panel discussion from individuals representing cohorts from low resource settings and low middle income countries. And what we heard from them was the need to really have a voice and to be an integral part of our teams, to have opportunities to lead in the science and to be participants in publications. We learned that, not surprisingly, they need help in terms of data science and having access to analysis pipelines. There's a desperate need for biobanking infrastructure and the development of best practices. And if given the opportunity, many of our LMIC cohort leaders would like to see their samples converted into data, and perhaps the IHCC can play a role in coordinating that activity. The other things that were mentioned was opportunities for training and cross-training, the ability to get IHCC to help with industry collaborations and negotiating legal agreements, and also to help with a cohort design, study design. And then today, the third day of our program, hopefully you've all been here for most of the day. We had a very nice opening from Ambrose Wonkum talking about why we should be investing in African genomics to highlight genomic variation in African populations. He focused on ecology, equity, and diversity, and espoused a bold vision to sequence 3 million Africans. This is an incredible undertaking and of great importance that the IHCC hopefully will be able to help with. We also heard from Francis Collins, challenging the IHCC community to help understand the COVID-19 pandemic, its biology, the response to therapies and vaccines, and encourage the IHCC and GA4GH to chart a path to open science to address some of these topics. Earlier this morning or this afternoon, we heard from cohorts from around the globe and how they've pivoted to carry out research on this pandemic over the past 18 months. One thing that's obvious is that cohorts have the advantage of having data from participants both before, during, and after infection, which is a really unique component of the nature of cohorts. We heard about a focus on mental health and from the Brazilian cohorts, rural populations, mortality studies from South Africa, host genetic studies and polygenic risk scores for developing infection from Chile, large-scale biobanking studies linked to health registries and contributing to the epidemiologic findings and genetics of COVID-19 from Denmark, about behaviors in serology and high-risk groups from Canada, and about the use of important data portals to allow for open science and from the United States, as well as innovation in sampling using home blood sampling. And then lastly, in the last hour or so, we had a discussion around the IHCC's ability to participate in Sentinel networks for pandemics. And we heard about the importance of the cohorts to provide real-time data as a unique opportunity to monitor emerging infections and pandemics. We also were challenged to learn about the heterogeneity response to infection from this data. We were told that the cohorts can really change the paradigm of access to public health data. Multi-omic approaches would be important, and we heard over and over again that sample size and diversity really matters. Having access to participants of the type that are in the IHCC cohorts is a real opportunity to rapidly get data and rapidly get samples that will lead to hopefully the better development of more accurate diagnostics for pandemics in the future. And then lastly, we heard that there's an absolute need to drive efficiencies in diversity and recruitment. So I really appreciate the last panel's insights as to what the IHCC opportunity might be in developing a Sentinel network, and there's really a lot for us to consider. So going forward, we'll be putting a meeting summary together. The working groups have their charges and will carry out many of the actions that came out of the breakouts and meetings that took place over the last 3 days. So in closing, I really want to thank our outstanding team for putting together a great meeting. Ida Donner, Carly Sullivan, Hannah Cannell, Ludi Dementon, all did a fantastic job. And as you leave this session today, I just would like you to remember the IHCC's new tagline, which is linking cohorts, understanding biology, and improving health. Thank you, and thanks for a great meeting, and have a great rest of your day.